Any questions in the room? And you might like to check chat to see if there's any questions coming from the Zoom in the chat. Okay, any questions in the room?
Just a question for the group on what's the outlook for clinical trials in asymptomatic mutation carriers versus in symptomatic patients?
How about now? Great. So the question was, what's the outlook for trials in asymptomatic at-risk people versus symptomatic patients? This is a question very dear to my heart because my wife is an asymptomatic at-risk and we really want her to be on drug. It's all going to depend on the safety profile of the drugs as assessed in animal studies. So in every country in the world, regulators make you do a lot of animal testing before you can ever expose a human to a drug. And the results of that animal testing determine how high you can dose and what kind of patients you can dose. For our divalent siRNA that our lab developed, we asked FDA for permission to dose both symptomatic patients and asymptomatic at risk. And our toxicology studies were very clean. We've made the whole reports available online. We went up to 8 times the dose that we plan to administer in humans, and we saw no adverse effects. So it was actually very clean toxicology. The problem is our drug is not active in the species. So we're looking at off-target toxicity, but we weren't assessing the effects of prion protein lowering. Now, I just told you that we have a lot of evidence that prion protein lowering should be safe. I believe that. But apparently we didn't convince FDA because they told us that we could dose symptomatic patients, but they wanted a monkey study before we could dose pre-symptomatic. So that's where we're stuck right now. And we do plan to continue discussing that with the agency. And I hope that there could still be a path forward to pre-symptomatic. But I think I probably speak for all of us. You guys can feedback. I think I probably speak for all of us when I say we all agree the highest and best good is to fully prevent the disease in people at risk, and we want to get there. It's a question of how we stage it, what kind of evidence we need to convince ourselves and convince regulators that it's safe to try. And then once we've tried it, what kind of evidence do we need to convince ourselves that it's effective and that we could chronically dose people? And of course, the last thing we want to do is harm someone who could have 10 or 20 healthy years left if we didn't treat them. Would others like to add?
Yeah, I think ultimately it comes down to risk benefit, and every drug has the potential to help and also the potential to harm. And at different stages of the disease, that equation changes. And for pre-symptomatic carriers that could be decades away from symptom onset, that's a very different risk-benefit equation to dosing symptomatic patients. And so it's a longer process to get there. And I wanted to highlight the work that the MRC prion unit in the UK are doing to look at CSF RT-QuIC tests in asymptomatic carriers over time and finding that patients are converting to RT-QuIC positivity years before symptom onset. And I think that is such important work because that is a candidate pool of patients that could really benefit from a pre-symptomatic treatment at that point. And there you know that they've converted, they have a high probability of developing symptoms in the next few years. And I think that type of work is really important to help us get to that patient population.
Any other questions?
Hello, my name is Saima Zafar. I'm from Germany. So my question is one of you is working on transcriptional level and one is at translational level. Yes. So what do you think if we can combine the both therapies and at transcriptional level, we are not sure that if we can reduce the with siRNA, we cannot go for 100%. So we can reduce some levels at that level and then join at translational level, because at proteasomal degradations and all these things might it might give some good evidence and things. This is just an idea as a biologist. Why not?
Yeah, the mechanisms are orthogonal and should be additive. So that there's no reason why combining them wouldn't lead to more profound lowering of PRP in more regions of the brain. Absolutely. I think ultimately you then get into questions of, will payers accept two super expensive therapies on top of one another? How do you test that clinically? All of those questions come up, but in theory, scientifically, absolutely.
Yeah, and I was just going to say that, yeah, I think it's a really neat idea. I think that it just also highlights like the importance of pursuing, because obviously we have to start with, monotherapies first, and it's the importance of pursuing multiple avenues at the same time. And yeah, I think there's precedent in other diseases where, you maybe start with monotherapies, but then move on to combo therapies as the drug development field matures. So yeah, I think anything's possible. We're really, I think, at the start of something, yeah, exciting.
We go across to Ayrton. I think he's got some questions. Have you got questions
Yes.
from the Zoom? Right.
Yes, I have two questions here. One is for everyone, I believe. Actually, both are for everyone. It's like, how is the process to enroll patients? So I think that everyone wants to know, if I have a patient, if I have someone that is symptomatic or asymptomatic, how can I enroll this patient in the future clinical trials? This is the first question.
Yeah, so I can start with that. And sorry, I was remiss. What I should have put up as I was talking was our posting on what's called clinicaltrials.gov. So this is, I think, a really important resource, not just for any prospective participants in the United States, but around the world, where it's a central repository for clinical trials, and it is a source of truth for like what is happening with that clinical trial at that time. I encourage folks to go there. At least at Ionis, that's where we, that's where we update our public information about the trial, whether we're recruiting or we're not recruiting, things like that, whether the study is amended. So honestly, I think that is the resource, and we partner with the patient advocacy groups to share updates, which they often refer back to clinicaltrials.gov for. So I don't know if folks want to add anything to that, but I think that's a major resource, right?
I would agree. I think that's sort of the best resource if you're coming from actually anywhere in the world. Clinicaltrials.gov, it's updated fairly frequently, so I would look there.
I would just add that I think we're lucky in prion disease to have really active patient advocacy organizations, and I expect that Debbie and Suzanne and Dina, that everybody will be sending out announcements. We're all really excited for clinical trials to launch, and I think that when they do, if you are on their mailing lists or you check our websites, you will find this information. We're really committed to making sure everyone knows.
Ayrton, perhaps you could type in the chat clinicaltrials.gov website so people can translate that and copy that link. That would be great.
Yes. So we have another question. So, it's okay to do another question?
Yes.
Okay, the another question is if there is a priority to enroll the patient according to the stage of this disease.
So I just want to make sure I heard that question correctly. So are you saying, is there a priority to enroll early stage patients?
Yeah, if there is some priority, if you in the clinical trial you enroll the patient that is in the mild or severe or asymptomatic symptoms, I believe that this is the question.
Yeah, and I can speak for Ionis's experience with our clinical trials so far. But yeah, I mean, so if you go on clinicaltrials.gov, one of our key inclusion criteria is to have early stage disease. And I think really one thing that we learned in conducting this trial was finding a balance between enrolling a population that was early enough that they could travel to clinical trial sites and meaningfully participate in the study, while also not being too restrictive that you couldn't find the patients, right? And so I don't know that there's a magical threshold, but that's kind of the judgment call that I think most folks or clinical trials will have to make.
That was great, Rob. I just want to add one question you could ask is, well, why are we restricted to early stage patients? Because in my trial for divalent siRNA, we're doing the same thing. And the reason is really that we need to know if the drug works and we don't want to do harm. And I know it seems counterintuitive. You might say, well, the most advanced patients are desperate. They have nothing to lose. Why can't we treat them? But I'll give two points here. One is in our animal models, we have tested treatment at different time points. We tested early treatment, treatment after symptom onset, treatment after very advanced symptoms. And what we have found across every experiment is that we can only do a benefit if you have at least a month to live. The drug takes a while to kick in. Prion protein has a certain half-life where it turns over. So even if you lower it really potently right away, it still takes a month for the protein to go all the way down. And when we treat mice that only have 3 weeks left, it doesn't benefit them because there's no time for the drug to become fully active. So we can't even do the experiment of testing if these drugs work if we have patients who are going to progress to death within 3 weeks. But the second reason is I think there is a risk of harming people by keeping them alive in a very advanced stage. And I recently had to write the consent form for our clinical trial. And in the consent form, you advise people about the potential risks of the therapy. And some of those are, well, we did toxicology studies and here's the findings and prion protein, so on and so on. But one of the things that I chose to write in that consent form is there is a risk that this drug will keep you alive longer, but with no quality of life. And I think that would be a harm to patients. And I think that's a reason to focus on people early enough in disease that it would be a benefit if we could arrest the disease in that state. And I take the inspiration from my mother-in-law's case where she was very advanced by the time anyone even suggested that it might be prion disease. At that time, I would not have chosen to enroll her in a clinical trial.
So thank you very much. I'm a medical doctor from Vienna, Austria. I'm a neuropathologist, and I have 2 questions. My first question is to you. So you mentioned this type of a carrier, right? I was wondering if it's also degraded in the proteasome or if it causes some inflammation.
You're talking about when PRP can't enter the ER, whether it can accumulate and cause stress. Is that
No,
the question?
you mentioned it's like a carrier or something that...
Conveyor belt?
Yes, conveyor, sorry.
That was the analogy for the endoplasmic reticulum, which is the secretory pathway.
Yeah, but
that
it
goes
binds
out
to this molecule,
Yeah,
right? And
it
then
does.
they go together to the proteasome for degradation, right?
Prion then goes to the proteasome for
Only the-
degradation.
only the
Only prion.
Ah, okay. And
Yeah,
this
that's right.
small molecule, is it also then degraded or?
It does turn over time. When you administer a drug, it doesn't stay forever in the body. Small molecules tend to be cleared pretty quickly, which is why you often have to take a pill once a day or twice a day even. The molecule doesn't last all of that long in the body. But I think what you're getting at is, does the molecule binding to the entrance to the secretory pathway cause some kind of stress in the cell? And the answer is no. So we've looked extensively at cell stress. We've looked extensively at what other things is this molecule doing other than eliminating PRP. And we find it's very selective and it doesn't cause any additional stress to the cell. Yeah. And we would definitely find that out in longer term toxicology studies as well, where you're administering to rats and dogs or monkeys for a long period of time. That's when you would also find problems, right? And toxicities. And so far looks good. So.
Thank you. And my second question would be about this is to all of you, about asymptomatic patients who, well, there is this question if they should receive these new kinds of therapies or not, because of course that there are the trials in the animals, and I mean, we know that the brain of a mouse is not like a human brain, right? And the cognition and everything is different. So when you disrupt PRP in a patient which is asymptomatic, would it cause some cognitive decline? Because, for example, if the person is, I don't know, intellectually more active, it would maybe be a burden, right? Because in the mice, there is this peripheral nerve problems, but you cannot test really the cognition in a mouse.
Yeah, I agree. So the question was whether we are worried about on-target toxicity in pre-symptomatic individuals, particularly with regards to cognitive function. And look, this is experimental medicine. There's always going to be risks. I can't say that the risk is zero, but we have studied this really, really hard, and we haven't found evidence that we should worry about it. I agree that it's very difficult to assess cognition in a mouse per se, but we've done lots of studies on the brains of mice. We slice them open. We look at their myelin. It's normal. We look at their proteomes, their metabolomes. My lab has done a lot of studies. And all the problems we see in the knockout mice are in peripheral nerve. They are not in the brain. The brain is very quiet. It looks the same in knockout mice as it does in wild type mice. Again, that's not a guarantee, but remember, first of all, we're up against a really terrible fatal disease and we can't afford to do nothing. So we are going to move ahead and take risks. Second of all, if you open your medicine cabinet and look in the drugs in there, lots of drugs in your medicine cabinet target proteins or genes that are essential, right? So things like even ibuprofen, right, targets a gene called COX-2, and if you knock it out, that's fatal, right? And yet we take it without even thinking about it, right? So just because there is some function to a protein and some negative consequence to having zero of that protein doesn't mean we can't make a safe and effective drug against that protein.
I think that what we can say is also that that's something that we're aware of and that we will be assessing cognitive function in any trial that we set up to try to make people educated about whether indeed there is a risk of some decrement or if things are stable. But right now we don't have that information. As we gain the information, it will be folded into informed consents, etc.
Yeah, and I was going to add on a similar point where it also highlights that the clinical trials are the experiments, right? To learn that, and participating in clinical research is you're taking a risk, and you learn from the clinical trials, right? And so it's an important question. It's a question that's being taken seriously. I agree with Erik that all the evidence that we know so far suggests that we're probably okay, but until we do the experiments, we won't know for sure.
I know Nina spoke about the presentation from the UK yesterday about RT Quick. Those that are on Zoom weren't here. So Erik, I don't know if you could summarize that. And my question to each of you is, will each of your approaches benefit from that so that if we do detect it early before symptoms start, will each of your drug therapies align with that and give us the best benefit of prevention or really slowing it down before anyone, at least some genetic families, are symptomatic?
So the study that was mentioned earlier was from Zehel Mock at MRC Prion Unit in London. And he had combined data from a natural history study on asymptomatic at-risk E200K mutation carriers in London, along with data from our study, similar study in Boston, one in San Francisco, and one in Tel Aviv. And between all of these studies, we had seen 8 individuals with the E200K mutation from whom we had spinal fluid, CSF, before they were sick. And the prion seeding was positive. RT-QuIC was positive. We could tell that they had prions in their brains. And then sure enough, they went on to develop disease. And in some large number, I think it was like 150 people with E200K who remained negative for prion seeding, none of them have developed disease. So as of right now, we have a perfect separation where it looks like if your prion seeding turns positive, that means you're extremely likely to develop disease in the next 1 to 5 years is kind of the range we think right now. So I think that's a really exciting research finding to be able to predict people's onset like that. It also has important limitations. So one is that we think that maybe the greatest good we can do is to stop the initiation of the disease even before your prion seeding is positive. So I wouldn't prefer to limit us to treating people who are already seeding positive, because that may mean we can't do as much good as if we treated early and prevented RT-QuIC from turning positive in the first place. The second limitation is that currently RT-QuIC only works for E200K. That's about half of mutation carriers, but the other half, which is mostly D178N and P102L, we don't currently have an RT-QuIC assay that works well for them. And so we don't have any way to predict their onset. And then maybe the third limitation I should say is that, look, that took decades of collecting samples at 4 cities across the world to get 8 individuals where we saw the seeding was positive. That's not a large cohort, right? We need to do clinical trials in more people than just that. So when I think about how we do trials in asymptomatic people, I think we need to include everybody who knows that they are at risk because they've gotten genetic testing and they have a high penetrance, high risk mutation, and maybe is above some minimum age cutoff relative to their mutation and when their risk starts to increase. And I think in that population, we should look at, is the drug safe and does it lower prion protein levels? Those are really important questions we need to answer more broadly for the genetic population. I think in the size and duration of a clinical trial, you might find 1 or 2 people whose prion seeding is positive, and then you could study specifically in them how the drug works. But that will be an anecdote, right? We're not going to do statistics on 1 or 2 people that's going to convince FDA this drug works. I think we still need a plan to test the safety and potency of these drugs in all pre-symptomatic people, whether or not their prion seeding is positive. And if it looks good in the couple of people who have prion seeding, maybe that's a story that we add on. We say, and look at this. This also looks promising. And maybe that helps the drug get to an approval if those data look good. But I wouldn't prefer to put all of my eggs in the basket of looking only at those people. Do you guys agree, disagree?
Yeah, no, I mean, I think it's undoubtedly an important question in the field. Are there measurable signs or something you can measure prior to symptom onset that could be some kind of starting criteria for someone that's at risk for developing genetic prion disease? And it's something that, you know, there's multiple studies going on to pursue that. And but I kind of agree where it's like, I almost think of it as a different population, right? Like, and as Nina was saying earlier, it's kind of similar as you kind of progress along that continuum of developing disease. It's like you kind of cross another threshold at that point, and the risk benefit to the individual person might also change in what they're willing to do. I can envision, at the individual level, willing to take an intrathecal administration for decades, right? And then some people might want to wait till there is some kind of sign that they can. So I don't know, but either way, I think it's early days. We need to keep exploring it. But right now, there's prevention as a population, and then there's treatment in this active population. And I think we need to just keep filling in that gap with these natural history studies. They're important.
sometimes once you've shown that you actually are benefiting people who are symptomatic, then it becomes easier and easier to convince people that you want to go into that window before people develop symptoms. And Ionis knows this from the SMA population, among others. But if you think about what happened with SMA and Spinraza, first being able to show, look, you can do a definite benefit in symptomatic people or early symptomatic people. Now let's see if we can treat people pre-symptomatically. It became a much easier argument to have with regulators. People could see.
I think we have one more question in the room. Do you have any more via Zoom, Eriton?
No more question here.
Okay, we've got one more in the room, and then I think we better move on.
Hello, I'm Ines and I'm from Germany. I'm a neurologist and I also do some prion research in the group of Inga. And I would like to ask you, probably Erik is able to answer this question. We know that in prion disease patients, the total PRP will decrease along the disease process. And if you see the same progress in mice, and if you are able to see a significant PRP decrease using any of the drugs you're using, and distinguish it from the normal disease progression. And I mean, also in different types of prionic disease, the profiles of the total PRP are different in terms of quantities, and if that has any effect in the way that the drug might influence the PRP levels according to the mutation or to sporadic.
Thanks for the question. So just to provide background, the question here is, we have cross-sectional evidence that PRP, not in the brain, but in CSF, prion protein in spinal fluid or CSF is lower in prion disease patients than in patients with other neurological diagnoses. And so that has suggested to us that maybe PRP release into CSF goes down as a result of disease process. It actually goes up in the brain. So we think that the PRP is incorporated into plaques in the brain. Maybe it's less soluble, and so it doesn't get released into the spinal fluid. Now, there's never been longitudinal data, right? Because historically, there was no therapy, there was no hope. Once you got diagnosed, you just went to hospice, right? Nobody was saying, after you're diagnosed, come in every month and we'll do another CSF and check how your prion protein is moving. And for years, we also did not know how we had to handle the spinal fluid in order to measure PRP. So it turns out PRP is a really annoying, terrible protein for more than one reason. It sticks to plastic. And so if you collect spinal fluid into plastic tubes, it sticks to the plastic and it's hard to measure how much of it was there. And so in my lab, we studied this and we figured out that it's as simple as add a bit of detergent and be careful how much plastic the protein touches as you go from step to step in your analysis. And so now we can measure prion protein reliably. And we've shown in pre-symptomatic people, they come in every year to our study in Boston. Over and over again, they have the same prion protein level. And each person's level is different. It depends in part on their mutation. But over the years, it's the same in pre-symptomatic, which is one reason it would be really nice to do pre-symptomatic trials. It would be really clear to show that we lowered the protein. Now, in symptomatic patients, we suspect that the protein in CSF is going down over time, but we've never had the perfect data to characterize that. So how are we handling that risk? I'll give a few different answers. I think Ionis was first to launch a human trial, and I think that this, I can't put words in your mouth, but if I was you, I think this is probably part of the reason why you have a placebo group in your trial, is that you need to assess what is that longitudinal progression of prion protein in CSF in the patients who aren't getting a drug. Another answer is we can do natural history studies on this. We just didn't think of it soon enough. So in Boston, a few months ago, we launched a new longitudinal observational study where we are asking newly diagnosed patients to come in and undergo a lumbar puncture to get spinal fluid at day 0, right after they're diagnosed, and then 4 weeks later and 8 weeks later. And we want to use that to figure out how does the prion protein drop over time. But that's a brand new study. We don't have those data yet. We also have an observational arm of our divalent siRNA trial, and we're hoping through that to learn some of the same answers. So Ionis is going to be learning about this. We're going to be learning about this. These are one of the risks in how we design trials, and that's one of the reasons why I call on my collaborators up here to share individual data from trials, because that'll help us all learn the answer to these questions. As for whether this is a problem that is going to make it impossible to learn anything from trials, I'm not that worried yet, right? There's a range of what the answer could end up being, but I'm optimistic because I think all of the types of drugs we've talked about up here work relatively quickly, whereas prion disease is a progressive disorder. And yes, the clinical progression can be rapid, but you saw from those data that Muck showed that the prion seeding has been there for years before you're symptomatic. And so I think a drug will become fully active within 1 month. And hopefully you will see a really dramatic sudden drop in PRP when we treat with a drug. Whereas my guess is that the decrease in CSF PRP from disease progression will be more gradual, and we will be able to distinguish it just based on the kinetics and the magnitude of change. But that's a hypothesis. We don't have the data yet. We're working as hard as we can to get those data. To add, would you like to contradict me?
No, I think you covered it. You didn't put word going up.
Awesome.
A rationale.
Okay, so thank you very much.